we want to try and get all the seats occupied. So we're going to begin our company presentations now, and I'm bending over here a little bit because we want to take the mic out of the stand. My name is Maury Ruffin with the Alliance for Regenerative Medicine. We've got a number of company presentations over the course of the next hour, and so we're going to begin with our first presentation, which is with the company Organovo and Keith Murphy, their Chief Executive Officer. Organovo is located right here in San Diego. Keith?
Thanks, Maury. So I'm going to say just a word as your partnering sponsor. Organovo is sponsoring the partnering at the event. And I'm going to lead in then and let Eric David, our Chief Scientific Officer, give his presentation for the bulk of the time. But what I wanted to comment is that when we started Organovo about 5 years ago in theory, and we operationalized it and kicked it off about a year and a half after that, I made a leap from pharma, from biotech, from Amgen into this industry on the basis of a hypothesis that this field was hitting its stride, was just growing, and really starting to hit the point where we were going to see the level of activity that we now see in the room. So I think the success of this investor and partnering forum, and I speak also as an active member of the Alliance for Regenerative Medicine, and ARM is taking a strong role in that with CIRM to help it move it along. I think the success of this meeting is evidence that hypothesis has been validated, that we are seeing these things happen. And Organovo remains very interested in partnering. Most people will know us, and you'll see Eric's discussion of our bioprinting platform, but we see ourselves as much broader than that, and we, more importantly, have built the team. Eric is a member of that team, Sharon Presnell, our Chief Technology Officer, and Mike Renard, our Executive Vice President of Commercial Operations. We built the executive team to really make great decisions in this space and work with good partners with new technologies. Bioprinting is an aspect of what we do, but we really see ourselves as a functional human tissues company. And there are so many aspects and technologies that underlie that we need access to through great partners. So I encourage, on behalf of ARM, I encourage everyone to take active advantage of the partnering opportunities that are made available here. And on behalf of Organovo, I urge you to look up us, find one of our executive team or scientists who are here, and join us in conversation with your great ideas about how we can benefit together. And I'll let Eric talk to you now about Organovo's technology in the company.
Great, thanks, Keith. Hi, everyone. As Keith said, I'm Eric David. I'm one of the co-founders of the company, along with Keith, and serving right now as the Chief Strategy Officer. I wanted to start today, since Organovo is one of the sponsors, as Keith said, and since we're the first company presentation, I thought perhaps I'd spend a little bit at the beginning and the end of the talk with a little bit of a broad industry perspective. So before I came on board full-time at Organovo, I worked as a consultant to pharmacos, biotech, private equity firms. And one of the main things I focused on was acquisition targets, due diligence, looking at new technologies. And I have to say, over the 7 years or so that I did that, the things that the people in this room do, tissue engineering, regenerative medicine, cell-based therapies, were absolutely the most interesting things that came across the transom. I mean, they just things that absolutely fundamentally constituted a paradigm shift in the industry. But no one really gets them. No one, or at least at the time, no one got them, and that wasn't that long ago. And that just underscores the fact that creating a new language, creating a paradigm shift, is really hard. And our industry is a slow-moving industry, and so we don't have that much experience with fundamental paradigm shifts. And so where can you turn for advice on something like that? Well, there is at least one industry on this planet that has a genuine fundamental paradigm shift pretty much every year or every 2 years, and that's the fashion industry. They absolutely turn 360 degrees every year or so. So Anna Wintour, the much celebrated, much maligned editor-in-chief of Vogue, the woman, for those of you who don't know, on whom the meme character in Devil Wears Prada was based. She said, look, it's all about timing. If it's too soon, no one understands. And if it's too late, everyone has forgotten. And it's absolutely true. Do you guys remember when there was the whole craze around gene therapy and every pharmaco had to acquire its gene therapy compounds? Do you remember the craze around aptamers? Do you remember the craze around cancer vaccines? Right, when the timing is off, we all suffer. And And so when I say that a lot of very smart people in this industry don't get what we're doing, I certainly think that was borne out in the last panel. But before I came to Organovo, one of the last things I did in consulting was I did a lot of interviews around new technologies. And there were a few different quotes that came up I want to share with you guys here. So this was just in 2011, an R&D exec who said, listen, this industry is about a decade from doing anything useful. Someone else, this guy was very emphatic. He said, can someone please explain to me the difference between cell therapy, tissue engineering, stem cells, and regenerative medicine? It's a dean of a top academic medical center who basically commented, every time I talk about stem cells, people still think we're working directly with human embryos. And then a strategy exec at a top 10 pharmaco who also just said, look, it's, this stuff just isn't for us. I think it's a lot of hype. So why do I put these quotes up here? It's certainly not to scare everyone, but to say, look, this is really hard. And we as a community of entrepreneurs and investors and researchers, the burden is on us to create this language together. And I think this particular part of our industry is really unusual in that it is and has been for some time unusually collaborative. And I just want to set that tone going into the talk, going into the meeting, that the burden's on all of us to come together and articulate what that language is and what the business models are. So let me tell you about our company. Organovo was founded in 2007 based on technology that came out of the University of Missouri. As Keith said, it's a bioprinting technology, and we are not so focused on selling the printers per se, but on creating tissues that come out of our printers. And those are three-dimensional, architecturally correct, fully human tissues. It's something that our industry has never had access to before. I'll tell you a little bit more in detail, but we're partnered with some major pharmacos, including Pfizer and United Therapeutics, so doing interesting things. We get, we've gotten some interesting coverage in the media. If you've seen some of that coverage and have questions, I encourage you to come talk to us about it. It was most recently we were covered in a nice article in the Wall Street Journal just last month. So what is 3D bioprinting? Because you hear the name and it sounds like kind of intuitively obvious. Obviously, I wish it were as simple as and as plug and play as a Hewlett-Packard printer, but it's getting there, right? And the key thing is you start with a cell ink, and that ink is a suspension of cells and prints individual droplets that each droplet contains about 10 to the 3, 10 to the 4 cells. There can be multiple cell types in the ink. And the printer lays down the ink with about roughly 20 micron precision, lays down these droplets. The droplets then fuse to form greater structures. And what you see at the bottom here is these droplets that were deposited in these cylinders, fusing together to form a vessel. Now, the technology that was developed by Gabor Forgax, upon which all this is founded, Gabor was fundamentally he was a biophysicist, and he was looking at human embryonic development. And he saw how, in chick embryo development, there were cell aggregates, droplets, that fused together to form greater structures. And so that's really where all of this came from. The observation that you let the cells do the work. What you want to focus on to get true architecturally correct end products is just letting the cells do the work themselves. And so it's all about how you put the cells together. You see them coming together, fusing over time in this series of images. And also, very importantly, it's also about the surface that you're laying the cells down on, because we lay down on a hydrogel that's specially formulated so that you allow the cells to interact with each other, but not with the surface. And that's critical, and it's a real point of departure from a lot of other cell-based technologies that you'll see. So we can print a variety of different architectures. As you've seen, I keep hitting the back button instead of the laser button. Sorry about that. You see cylinders. You see patch here. These are cardiac myocytes in this bioink that that are coming together to form a patch. So we can print a variety of different geometries. And what do you use this for? What is the technology for? Well, obviously a couple of different applications. First of all, in drug discovery, we can print into 96-well plates, or we can print larger tissue blocks to create disease models for diseases which, let's face it, there are a lot of diseases out there that have very poor disease models in pharma. And for ADME and tox. But the critical thing is this really fills a very unique gap that's never been filled before between 2D cell culture models and animal models on the one hand, and then clinical trials on the other hand, right? I mean, this is real, fully human, dynamic, living tissue that you have access to in discovery and preclinical. And then obviously the other focus here is on therapies. And obviously, eventually, the goal would be whole organs for transplantation, but on the way there, patches and organoids and other sorts of things that we can do. So let me say a word about each of those areas of focus. On the drug discovery side, again, when I was working with R&D organizations as a consultant, the mantra was really, how do we learn to kill drugs faster and cheaper, or greenlight them with more confidence faster and cheaper? Because, I mean, look, even just over the last year and a half, there have been, off the top of my head, 7 very high-profile programs that failed in late-stage clinical trials for either safety or efficacy, right? That should not be happening. That is a $600 million way to fail, right? You want to fail much earlier than that. And again, having access to fully human, 3D architecturally correct tissue is critical for that. On the tissue side, you can create patches, you can create small organoids. Obviously, it is a longer road to create an entire organ. And we can create different bits and pieces now, but there are lots of steps that can be commercialized on the way there. So we've been working with a number of partners, the ones that we publicly discuss, Pfizer and United Therapeutics on the company side, and then we have one of our bioprinters right here at the Sanford Consortium for Regenerative Medicine, and I think there'll be a tour and a little viewing of the printer tomorrow. Going to the Harvard Medical School, Wake Forest, and always looking for new partners to do interesting things with on the academic side and, of course, on the industry side. I want to underscore just a few benefits of the platform, and then I'll show you some examples of tissues that we've printed. So again, our tissues are 100% cellular and without a scaffold. While we do print onto a hydrogel, as I said, that hydrogel is later stripped off, removed. And so the constructs that we make are, they're just the cells and whatever extracellular matrix the cells are making. And that's critical, right? Because when your body makes tissue, it does not print out a scaffold and then put the cells onto the scaffold. That's not the way our bodies work. And therefore, it's not the environment that the cells are used to. Likewise, our technology also enables us to print multicellular tissues and put those cells in correct architecture. Again, why do we think that 2D cell culture, for example, is a poor model? Well, when was the last time you saw a tissue in the body that was made of a single cell type, much less in 2 dimensions? It just is. It's not what these cells do. It's not what they know how to do. So the technology enables us to do things with these tissues that really do mimic the in vivo environment much better. And so you see things that you do not see in other forms of technology. So genuinely, genuine compartments in the tissue comprising different functions, as you see in these images of bioprinted liver. Having a genuine barrier and transport functions with an epithelial layer on top and stromal layers beneath. And then very importantly, you see microarchitectural structures forming. So in some of these, in the bioprinted muscle and liver, you see some microvascular networks forming here. We see tight junctions forming between cells, which you obviously do not see in 2D cell culture and a number of other technologies. So really, things that very much echo the in vivo environment. This slide, I think I've covered most of this, so I won't dwell on that slide. So what have we printed is a question we often get asked. Obviously we are a small company. We have limited bandwidth. We try to expand that bandwidth through partnerships. But you see on this slide the things that we have tried to focus on. Certainly, if there are other tissues of interest, please come talk to us if any of these are of interest. Obviously, we'd be happy to talk about them as well. I'm just going to show you some slightly more detailed examples of some of these. So one thing that we've really tried to focus on, one thing that we've noticed about our partners is people come to us saying, okay, well, in particular, we're interested in disease models of areas where the current disease models are very poor. So a lot of the fibrotic diseases. So this is an example of pulmonary fibrosis and an IL-13 inducible pulmonary fibrosis model that we built. You see the control tissues up here, and then induced with IL-13. You see the nice epithelial layer on top, the layer of fibroblasts and stroma down here. You see down here some of the fibroblasts having taken on more of a smooth muscle phenotype, and you see some of the microvascular networks in here. Bone, another thing that traditionally pharma has not had good models of, because either they have animal models, or if they want to do human tests, they end up with cadaveric bone, and there's not much you can do with cadaveric bone. So this is bioprinted bone that has that full staining for alizarin red and alk-phos, osteopontin. This was this tissue was crunchy when it went on the microtome. It actually crunched the way bone does. This is human vessel that we built. You see obviously the endothelial layer, and then you have the layer of fibroblast, layer of smooth muscle. So the same layers that you see in traditional vessels. I'm rushing through for the sake of time, but one thing that's very impressive about this, you build these tissues and then they mature over time in a bioreactor, and you see by day 3, you see that tissue develop a burst pressure on the order of 315 millimeters of mercury. So again, by that point, it's got a burst pressure higher than any hypertensive crisis that you see in patients. By day 21 or so, you see burst pressures of almost 800 millimeters of mercury. If you were to take a human vessel out of the body and measure its burst pressure, it's generally the highs that you see are around 1,000, 1,200 millimeters of mercury. So you really see the tissues mature to in vivo kinds of strengths. I'm going to close in just a minute, and in closing, I just want to share a very quick story with you guys. So very quickly, does anyone know who this is? Yell it out. Ernest Hemingway. Usually people say either Obi-Wan Kenobi or Craig Ventnor. And I have to say to Dr. Ventnor, doesn't look anything like you, but to be confused for Obi-Wan Kenobi or Ernest Hemingway, probably not a bad thing. So why do I put a picture of Ernest Hemingway up here? Well, so let me tell you a story. Picture the scene, right? 1920s Paris, dark rainy night in the back of a bar. Ernest Hemingway is sitting there at a table with Gertrude Stein, and James Joyce, Ezra Pound, F. Scott Fitzgerald, right, these luminary writers. And Gertrude Stein slaps a $100 bill on the table and says, that is for the person who can write a novel in 10 words or less. Now, I have to take us for just 1 second. My wife's a writer, and you want to talk about an industry with a funding problem. You think we have it bad in biotech? Try fiction. So when my wife tells a story she likes to see, yeah, that was probably the last time a group of writers saw a $100 bill. So anyways, they're sitting around the table, and Gertrude Stein challenges them to write a novel in 10 words or less. And so people laugh, what a ridiculous challenge. But of course, Hemingway grabs his napkin, grabs his pen, scribbles something on the paper. He then slams the paper down on the table, grabs the $100 bill, and walks confidently out of the bar. So what was written on the paper? 6 words. For sale, baby shoes, never worn. Right? It's heartbreaking. It's a complete novel in 6 words. It's got a beginning, a middle, and an end. It's got all the subtlety and ambiguity of a novel. So why am I telling you this story? Well, look, no one in this room is naive enough to think that creating tissues from scratch, replicating functions of the human body is easy. We've all been trying to do this for a long time. But first of all, the good news is, at least at first, you don't have to recreate the entire organ. You don't have to write the 800-page novel. Sometimes 6 words suffices if it's the right 6 words, if it's the right cell types, if you've got the right functional assays, you're looking at the right endpoints. And I think that's critical for all of us to focus on. Second reason I bring it up is that was a time in Paris with that group of writers that was unusually collaborative in literature. It was a group of people, they all sat there reading each other's manuscripts, giving each other advice, and together they created 20th century literature. So again, I come back to this theme of we as a community have to really collaborate to bring these things together, to articulate how we're going to get them done. Because right now, everyone has a piece of the puzzle. Some people have the cellular inputs. Some people have the technology to lay down the architecture. Some people have the functional assays. We need to come together and figure this all out together. So that's why we're here. I thank you for your time.